Hi everyone, welcome to my channel Pure Biology, and today I come with another video related to immunology, that is the antigen antibody interaction. Okay, this is very short and brief topic related to immunology. And before going to this topic, I want to say thank you to all of you who have already subscribed my channel, and the newcomers, I request to the newcomers in my channel, obviously you share my channel with your friends if it is helpful to you. Okay. uh, I have already uploaded different topics related to immunology and other obviously topics so in my channel, and you just check out that all. Okay. And if it is helpful to you, then obviously subscribe my channel. Okay, so let's continue. Antigen antibody interaction. What is called the antigen antibody interaction? Antigen antibody interaction means the binding of antigen with the antibody, and this is a very specific. Okay, this is very specific. The binding between Ag and Ab, this is actually occur due to the non-covalent interaction. Okay, weak non-covalent interaction, and this interaction may be possible by some forces. These are van der Waals weak forces, electrostatic forces, and hydrophobic forces. Okay, these three forces are generally possible. Okay, so this binding of Ag and Ab is possible by the three type of forces. One is the van der Waals weak forces, electrostatic forces, and the hydrophobic forces, and obviously the binding is very reversible and weak. Generally AgAb binding is weak and reversible. Okay, and the smallest unit of Ag that is capable of binding with the Ab means antibody, so this is called the epitopes. And the term epitopes I have already explained in my previous video, so check out obviously that term. And this is called the epitope. And the corresponding, generally the corresponding area on antibody that combining with the epitope is called the paratope. Okay. Then what is called the paratope? Where the antigen bind, the place of antibody where the antigen binds, this is called the paratope. Clear? So the epitope is what? The smallest unit of antigen by which it binds with the antibody. This is called the epitopes, and paratope is the area of the antibody that is actually responsible for binding with the antigen is called the paratope. Clear? And now, number of epitopes on the antigen surface, generally known as the valence. What is called? The number of epitopes that present in the antigen, that is called the valence. So, if one epitope is present in antigen, then it is called the monovalent. Clear? And if there are same type of epitopes present, many copies, okay, not only one copy, more than one copy is present, then it is called the polyvalent. Clear? So what is monovalent, what is polyvalent, what is valence, you all know about that. Next term is the affinity and avidity. What is called the affinity? Affinity actually the intrinsic association constant that characterizes the monovalent association, and this is called the affinity. That means the simple word is the binding of single antigen and single antibody is called the affinity. Okay, and the binding of single antigen and single antibody is very weak, so the binding of the antigen and antibody not remain for a long time. Okay, so what is that? The binds AB binds with the AG very weakly, and that dissociates easily. Okay, that dissociates easily. And high affinity, that is AB, binds to AG for a longer period of time. Okay, high affinity, this is low. Okay, this is high, HIGH, high. Here, this is high. The high affinity between AG and AB. The high affinity between AB and AG that remain, the binding of AG and AB that remain for a longer period of time. Okay, but the affinity becomes very low between single antigen and antibody, then the binding easily dissociate from each other, or AG easily dissociate from AB. That's for the weak binding. And when AG consists of many repeating epitopes, okay, many repeating epitopes, then it is called, we know what, that polyvalent or the multivalent. And obviously epitopes are identical or same type. Clear? And then the binding of AG and AB, that depends on the sum of affinities. Here only single antigen and single antibody bind to each other. That's why single affinity is needed here. But here many antigens, or you can say one antigen with the many epitopes, bind with the AB. Okay. So here the sum of affinity is very important for their binding process. Clear? And that's why here AB is also multivalent. For binding of different parts of epitopes, or the same type of epitopes that present in the multiple number in antigen. So that's why binding for antigen AB must multivalent. Okay, because AB, they bind all the epitopes of the antigen. So here the interaction between the multivalent AB and AG is known as the avidity. Clear? This is the affinity and avidity. And now we will learn about the cross reactivity. Okay? This is the cross reactivity. Okay, so this is the cross reactivity. Cross reactivity is what? Generally AG-AB is a specific, but sometimes AB elicits or elicits by one antigen can cross react with an unrelated antigen. Clear? That means antibody is very specific to antigen, but sometimes, sorry for the disturbance. Okay, what I'm saying, that antibody, they are very specific for a particular antigen, but in sometimes it has another capability that to bind with unrelated antigen. Okay, not the specific antigen, but it try to bind with the unrelated antigen. Okay, so in that situation the immunologic reaction is seen where the particular antibody or TCR that react with two or more antigens. Okay, that react with two or more antigens. In that situation, this is called the cross reaction. So what I am saying here, that antibody, these are very specific to antigen, but sometimes it tries to bind with the unrelated antigen. In that situation, the immunologic reaction where the TCR or antibody that binds or react with two or more antigens, not a specific antigen, including specific antigen obviously, but it tries to bind with unrelated antigen. Okay, in that situation the cross reaction is seen. And here, why this cross reaction? Because this is seen simultaneously. That means specifically it binds with antigen and obviously it binds with unrelated antigen. Okay, it is possible, or in sometimes it is not possible. The simultaneous reaction is not seen. This is seen individually. So this is called the cross reaction. Okay, the capability of the antibody to bind with two or more antigen. This is called the cross reaction. Okay. And this cross reaction is seen in another time where generally antigen they tries to bind the actual epitope site, okay, epitope site of the antigen. But this binding, that means the single epitope and the antibody complex, this is not more stable. And so sometimes what is seen there, the another epitope that is not identical, okay, that is not identical but structurally same like the first epitope. In such situation, the cross reaction is seen. Okay, actually antibody cannot understand the particular epitope here. So that's why it tries to bind with the epitope that is not actual antigen's epitope, but structural similarity is there. So that's why antibody cannot recognize the actual epitope. So it tries to bind the another epitope of the antigen which is similar or structural resemblance with the first epitope. Okay, that is actually antibody has to recognize. Okay, so this is called the cross reaction. Okay, or the two situation where we can see the cross reaction. Okay, so this is the actual affinity, avidity and cross reaction. I just draw very roughly what is the actual cross reactivity and affinity and avidity, just to understand easy way. Okay. Okay, so this is very rough structure of affinity and avidity and cross reactivity. Okay, here you can see the shear epitope. Both have the structural similarities, but here the two different epitopes. Okay, not different epitopes, a similar epitope. Okay, the structural resemblance is not seen here, but these are two similar epitopes, and here the structural resemblance is seen here, but these are not similar epitopes. So the cross reactivity is seen here, and this is the affinity where the single binding you can see, and this is the avidity where the two or more, so where it is the two structural binding I showed here. Okay, so this is the rough diagram of affinity, avidity and the cross reactivity. So the antigen antibody interaction topic is totally ended here. So I think you understood that, and please share this channel with your friends. Again I repeat, and for the growth of this channel, and continue with me.